Shall we start, Doctor?
Yes, please.
Good morning, students. Good morning to all. Today we are entering the day 19 of our BIIS-11 workshop. We have with us today Dr. Sarjeet Kaur. She is a principal scientist at the ICAR National Institute for Plant Biotechnology, New Delhi. Good morning, students. She has an MSc in biochemistry and PhD in biochemistry from Indian Agriculture Research Institute and was awarded IARI Gold Medal for her doctoral work. She also has postdoctoral training from Salk Institute, California and Ohio State University, Ohio, USA. She has 35 years of research and teaching experience in plant biotechnology and molecular biology. She is working in the area of isolation and characterization of novel genes from Basilar Stringency and has been group leader for the chickpea project at NIPB. She has guided several MSc and PhD students and has over 45 research publications. So with this short introduction, I invite Dr. Sarjeet Kaur to deliver her talk. The floor is yours.
Thank you, Dr. Murugan. So I will share my presentation. May I know, these are UG students, right?
Yes, UG students from life science and pharmacy.
Life sciences. So I hope you are able to see my screen.
Yes, doctor.
Okay. So I'll be talking on transgenic crops for biotic stress resistance. And this is the institute I work at, which is National Institute for Plant Biotechnology. Now, of all the biotic stresses in agricultural ecosystems, the insect pests, they cause the major damage. So there are other biotic stresses also, such as fungi, viruses, bacteria, weeds. But insect pests cause main damage, and that is also has been the focus of my research. So these insect pests, they belong to different orders. For example, in this, these are the beetles, which is the Coleopteran order, and these are the rest are the borers of different kinds, which are Lepidopteran. So let us first try to understand what chemical insecticides do to our environment. So for many people, the first introduction to disastrous consequences of use of these chemical pesticides, it came through this famous book, The Silent Spring by Rachel Carson in 1960. This was published, and it led to public awareness about over-reliance on chemical pesticides, which has such a detrimental effect on our environment. And it also leads to development of resistance in pests towards these insecticides and eliminates the natural predators of pests. And so we can say that pesticides, though they are effective for short-term pest control, they have long-term detrimental effects. So they are an unsustainable strategy of insect pests. So then the thought developed that there should be integrated pest management strategy in which there should be different components, including the biocontrol. And for biocontrol, there are biopesticides, and of this, Bacillus thuringiensis is the main biopesticide. And this is a picture of Bacillus thuringiensis, which is a bacterium. And it has been the major biopesticide in agriculture, forestry, and mosquito control. And of the 1% market share of biopesticides in the total pesticide market, 95% of biopesticides are the BT. They are derived from BT. So Bacillus thuringiensis, a short form we call as BT. So it is an aerobic gram-positive bacterium, and it forms spores and also insecticidal proteins such as crystal, WIP. WIP is short for vegetative insecticidal proteins, cytolytic proteins, and secreted insecticidal proteins. And they control, these proteins are toxic to a wide range of target insects of different orders such as Lepidopteran, Dipteran, Coleopteran, and Hemipteran. And as such, this has been used for over 5 decades for control of insects. Now, the overall spectrum of a Bt strain will be either the additive or the synergistic function of how many individual insecticidal proteins it produces. And there are also other virulence vectors, but they do not have much commercial significance as of now. So this is a picture of the bacterium, and this is the spore. And during sporulation, it forms a crystal in which we have these crystal proteins. So dry proteins, we call them for short. So they are in these crystals, and their three-dimensional structure of these proteins is in the form of three domains, which you can see here, 1, 2, and 3. So we call them also as three-domain toxins. Now, a little bit about the history of Bacillus thuringiensis. So first of all, Professor Ishiwata discovered it in 1901 and from diseased silkworms, and it was known as Bacillus soto. He named it as that. But then subsequently, Berliner found it in a grain mill in Germany, district Thuringien, so it derived its name as Bacillus thuringiensis. And then the important thing is the discovery of this Kustaki strain in 1962 by Kustaki, and which is the major strain which is used in biopesticides. And in 1981, it was known that there are plasmids inside this bacterium which bear these insecticidal crystal proteins, and the first gene was isolated. And in 1991, the first Bt transgenic plant, that is, the plant having tri gene in it, was developed. So initially, when there were number of isolates was less, the classification of these Bt isolates isolated from different places worldwide was by serotyping, which is based on their H antigen serotypes, and they were named as such. And subsequently, and this is just to show the morphology of some of the main strains, that the crystal can be either in different shapes, either it can be bipyramidal, or it can be cuboid or amorphous, and it could either be outside exosporium or within the exosporium, and generally at the end of sporulation, but some and in other bacilli, the proteins are whatever would be produced, they are during the end of vegetative. Now, right now, there is a large variety of crystal proteins and also the vegetative insecticidal proteins. So, the nomenclature has been revised, and now we name them according to. Earlier, we were naming them based on sequence homology and insecticidal activity. So there were only 4 major classes. But then, as research has progressed from 1980s till now, the nomenclature has been revised, and they are given 4 ranks: primary, secondary, tertiary, and quaternary. So, for example, a particular gene named as Cry52AC1, this incidentally was isolated in our lab. And this is named based on percent sequence homology with other cry genes. So up to 45% first designation, up less than 78 second. So we will all call them Cry52A, and tertiary up to 95% sequence homology, and after 95%, that if they're more than 95% similar, we give them rank as Cry1 AC1, AC2, and so on. And similarly, we name the vegetative insects. So this picture shows the huge number of Cry and VIP and SIP type proteins which have been isolated. And right now, there are 78 primary rank Cry proteins, 4 VIP primary rank proteins, and 3 cytolytic rank proteins, and they are colored according to their toxicity. So, and this shows the toxicity of various types of cryotoxins to different hosts. So these are the different insect orders. So these proteins are toxic to Lepidoptera. Then we have a range of cryproteins toxic to dipterans, which are the flies kind of insects, and the coleopterans, which are beetles kind of insects, and hemiptera, which are aphids, and hymenoptera, and so on. And also, interestingly, some of the cryproteins, 31, 42, 45, and 46 have been found to have parasporins. Their proteins are named as parasporins, which are toxic to human cancer cells. So they have applications in medical science. So now coming to the structure of cryproteins, most of the cryproteins, as I said, they are three domain toxins. So X-ray crystallography was done to find out the structure of some of the proteins, and they were found to have these three domains. So the blue one is the first domain, which is, depending upon its function, now known to be the pore forming domain. It forms a pore inside the insect midgut membrane. And then the second one is domain 2, which is the receptor binding domain. So it binds to receptors in the midgut membrane. And the third one, which aids in this binding, is the domain. So they are, we can label them according to, starting from N terminal of the protein. About first 250 is domain 1, next to 300 is domain 2, and the remaining 150 amino acids is the domain 3. Now, there is sequence homology among cryproteins, and in all cryproteins, for example, 74 listed here, we are having these colored boxes which show conserved regions of sequence homology among all the proteins. And so, these are the regions which are common among all these proteins. And this implies that homologous proteins, they will have a similar kind of structure. So now this is a basic slide of showing how the Bt toxin acts, the cryprotein. The pH inside the Lepidopteran insects is alkaline in the midgut. And in this alkaline pH, when the crystal is ingested, it undergoes some degradation by protease. So the activated toxin is released, and this toxin then binds to the receptor in the midgut, and it leads to formation of a pore, and upon which there is subsequent cell death and septicemia. Now, this is the detailed mechanism of action at cellular level. So, the larvae eat these cryproteins, and they are solubilized, and protoxins are activated by midgut proteases, and then they bind to receptors in the apical microvilli. So, this is the midgut section shown here in electron micrograph. So, toxin binds here, and then it leads to formation of pore and septicemia and cell death. Now, on a molecular level, also this mode of action has been worked out, and different kinds of receptors such as alkaline phosphatase, aminopeptidases, and cadherin have been found out to be responsible for binding to these toxins in an oligomeric fashion. So this is monomer toxin, and then they form an oligomer, and they bind, and then they lead to pore formation. Similarly, the mechanism of these are the VIP proteins, and of interest to us are in yellow the VIP3 proteins, which are toxic to Lepidoptera, because as I said, most of the crop pests are majority are Lepidopteran insects. So these VIP3 proteins, they also act by pore formation, and they have also been transferred to cotton and maize in transgenic crops. So their mechanism of action is also by pore formation, and this is shown here. But the receptors are different from cryproteins, and they have instead of 3 domains, they have 5 domains. And now, since the receptors are different, this has an implication which I will discuss later. That it means that the cry and the VIP proteins, they will bind to different receptors in the insect maker. So if the insect develops resistance to cry proteins, it will still be susceptible to the VIP cry. Now we will talk a little bit about Bt-based biopesticides, because this was the first Bt-based control which came into picture, and transgenic plants, which came only in 1980s. So they have been used on over 50 crops since last more than 7 decades. And now most of these are based on this subspecies Woodstocky HD1 strain. But there are limitations with these, which are there is narrow host range, and the main limitation is there is low persistence in the field. We spray Bt pesticide, and there will be a wash off by rain, and also inactivation of these proteins by ultraviolet light, sunlight, heat, and they may react with leaf exudates, and they will not reach inside the plants. And so, because this is only surface application, so several sprays will be necessary. Also, there is high cost of production of these as compared with the chemical pesticides, which are much cheaper. So media optimization is required. So several improvements have actually been made, and this is an example of some of the natural strains which have been used, which have been developed as BT biopesticides, so mostly Kustaki and some Israelensis. So Israelensis is for bacterial flies, mosquitoes application, and and Coleopterium tenebrosus, which is for beetles. Now, the biotechnological improvements have focused on enhancement of insecticidal spectrum on plant resistance and increasing the insecticidal efficacy, molecular. Now, field persistence, how do we increase? We transfer these genes to the microbes which have better persistence, such as Bacillus megaterium or Pseudomonas fluorescens or root noculating bacteria or some endophytes, such as Clavibacter species. And also some improvements in formulation technology. So recombinant strains having desired combination of insecticidal proteins, that is more than one, with enhanced toxicity have also been engineered. But then what are again the limitations with these is that potential, so with any biotechnological product, we have to see its environmental impact. So the potential environmental consequences, they need to be assessed prior to commercialization. And safe will be that the BT strains which are through the natural, the process of conjugation, which naturally also occurs in nature. So despite ongoing research, now these kinds of transgenic microbes are still in their infancy. That is not very well commercialized. These are examples of some of the BT biopesticides which are having these novel recombinant strains. And this match I've highlighted here. Now this is the one which is the Pseudomonas fluorescens, which is having fly proteins from BTs. So now during 1974, can say the biotechnology revolution came because during this time, the restriction enzymes also came to be known, which can restrict DNA in a very specified way, and also became known the TI plasmid, which is a tumor-inducing plasmid carrying pDNA in soil bacterium, agrobacterium genes. And now this was used as a gene carrier to transfer genes of interest from another organism. So this is from agrobacterium patients, and we can insert DNA into this tDNA, make a recombinant plasmid, and introduce into the plant cell and get a transgenic. So this became possible to develop transgenic plants. Now, the advantage was that the problems of field application of BT biopesticides were overcome because the plant itself was producing these genes. And the first insect-tolerant BT transgenic plant was developed in 1987, and commercial release was in 1986. And not to use these, we use synthetic crygenes. Why? Because the bacterial crygenes, there is a concept of codon usage in all species in which you know genetic code and their code is degenerate. So there is a preference of codons for the same amino acid. In different species, different codon will be selected. So that is the basis of codon usage. Now to make it plant-proof, plant prefer codon usage because bacterial genomes are generally, they are GC low content, AT rich. BT is AT rich. So GC content in bacterial genome is only up to 37%. By GC, I mean guanosine and cytosine residues. So we have to increase that GC content. So we make synthetic genes. So they were introduced into plant. But only a few genes were used at that time. And several plants, transgenic plants such as maize, cotton, tomato, brinjal, rice, potato, soybean, they have been developed. Now, the one which has been commercialized in India is BT since 2002. And up till 2019, there has been 94% adoption rate. That is 94% of the type of cotton grown in India is BT cotton. and the area is about 11.9 million hectares. So this reflects the success of BT cotton in India. So this is just a quick slide to show that from BT, we get the toxin and we modify, we make it a synthetic to increase its TC content. By tissue culture, agrobacterium based, we transfer it to crop, develop the transgenic crop, and then it has resistance to this bollworm, which is the cotton bollworm, which is Helicoverpa armigera. And so we get increase in cotton growth. So these were the initial BT crops in the global market. Was mostly having these chiefs and mostly by Monsanto and Syngenta and Dow Agro, some of the private companies. Now this is the global scenario of transgenic crops, and there are 5 major countries: USA, Brazil, Argentina, Canada, and India. So they are having this much shares, and this is the respective share crop wise. So this is the biotic cotton, which is and this is again reflecting that the transgenic plants have been accepted, largely accepted globally. And the top 5 countries, as I said, they are USA, Brazil, Argentina, Canada, and India. And the BT genes have been transferred to cotton, soybean for lepidopteran pest, and to maize for lepidopteran and phylloxera pest. And interestingly, in BT brinjal eggplant in Bangladesh, it also contains this BT gene, and BT brinjal is growing in Bangladesh. Now, let us just have a look at the pre-BT cotton days. So when the smallholder cotton farmers were really suffering because of this cotton complex, pest complex, which is comprising of this major cotton bollworm, which is Helicoverpa armigera, pink bollworm, Pectinopora gossypiella, and also spotted bollworm, Ilias vitica. And the most damaging is Helicoverpa. And there was up to 30 to 35% of the crop was lost. Then there was introduction of BT cotton in India because in 1990s, Monsanto offered this transgenic technology public sector institutions in India. And it was in 2 forms. There was this Tri1AC and Tri1AB gene cloned in Escherichia coli, and also the transgenic cotton seeds of this cotton variety, Coca 312, which contained Tri1AC. So this was the first project, National Agricultural Technology Project, NATP. So I was a part of this project to develop insect resistance BT cotton. And the second initiative was by private sector, by this company, Maharashtra Hybrid Company, and Monsanto to develop BT cotton. So this is the BT cotton success story in India. So there is an approval committee, which we call as Genetic Engineering Approval Committee, GEAC, which granted permission for commercial cultivation to Mexico in 2002, 3 cotton hybrids, and after several years of field. So then there was rapid increase in it, and in this cotton paper 2017, it is said that there has been increase to 93.14 in cotton production, and the cotton production has raised from 7.6 in 2002 to 10.6 in 2016. And now India is the world's largest cotton producer and second largest exporter of cotton. And now currently there are more than 1,400 hybrids, transgenic hybrids of BT cotton suitable for different agroclimatic zones in India. Now, since transgenic crops have also been a subject of debate as to their environmental safety, I will spend some next few minutes on what are the perceived ecological and food safety risks with transgenic crops and how we can manage this. So it has been said that transgenic will have effect on phenotypic traits. For example, it will have what is the safety towards non-target organisms, what is any impact on environmental fitness of crop, increased weediness, is there any yield compensation, and any unpredicted effects such as any changes in level of secondary metabolites, and food safety. So all these are thoroughly ascertained before any commercial use. And several studies have been done, which are eco-toxicological studies, to show that there are no adverse effects or negligible risk of BT on non-target organisms. And also, it is assessed on a case-by-case basis, by which I mean that each transgenic crop before commercial release will go through this thorough environmental impact assessment. There will be laboratory tests and there will be small scale and large scale multi-locational field trials to a certain safety. And now one concern has been that whether there will be inflow in centers of origin of diversity, which is India is very biodiverse. So in those regions, and that is one reason why BT brinjal, after being developed as a transgenic crop, hasn't still been released, because some people have expressed a concern that India, there is a lot of eggplant biodiversity. What if there is gene escape to other varieties? Now, gene escape is very unlikely. It will be a rare event, so horizontal gene transfer is rare, and transgenic crops are more likely to transfer genes than the other crop cultivars. That is, as happens in nature, not likely to be more than that. And to tackle this also, the transgene are having strategies such as chemically induced transcript design. That is, expression will be induced only by spray of a particular chemical, or transferring the transgene to chloroplast. Chloroplast also has a genome, so transferring it to there, so that there will be maternal inheritance of this gene. That is, it will stop with the first generation. So regarding food safety also, the concern has been that there will be spread of antibiotic resistance, and there could be toxic proteins and allergens. So dry proteins do not contain sequences which are having any allergen. This has been found out by molecular modeling studies and bioinformatics studies, and concentration is also well below 0.1%, and there is no evidence of toxicity or allergenicity of BT crops humans. And nutritional composition also remains unchanged. Food safety concerns are also negligible. And regarding gene escape into environment, other strategies such as elip of these antibiotics, which are antibiotic resistance markers, are used during selection of transgenics and or herbicide resistance markers. So there are strategies which eliminate this marker gene once the transgene has been developed, such as Krelox recombinase mediated marker gene acquisition. So there needs to be public awareness on the merits of transgenic. And for this, food labeling is done. So India do food labeling if it is genetically labeled, genetically modified food. So how the environmental risk assessment of BT crop system? So this is done based on certain protocols, and so this is requires the regulatory approval. And this is according to this Cartagena biosafety protocol, which was signed by different countries and countries. And post-market environmental monitoring is also done. And before commercialization, internationally accepted risk management strategy, risk assessment strategies are done, as I explained in the previous. Then on-farm follow-up monitoring is also done, that whether the required norms are being followed in cultivation, and strict implementation of these management strategies and regulatory regime. In India, we have a regulatory policy for transgenic crops, which is monitored by Department of Biotechnology. It is very much in place. And then now, since there has been some resistance to transgenic crops, the new innovative technologies such as genome editing are being used, which is CRISPR-Cas. You may have heard about it, so which is short for trusted regularly interspace short palindromic repeats, and CRISPR-associated protein line, which are now, the major concern with transgenic crops has been that since if it will be particularly a single gene, so which will be expressed generation after generation, so this will lead to increase in selection pressure in insects, and they will develop eventually resistance, like they develop resistance for some of the chemical pesticides. Now, this is an issue which needs to be addressed. And for this, the research is going on to know what are the genes responsible for development of resistance in insects. And luckily, these genes are the recessive alleles type. That is, there need to be two genes for the function to be expressed, two alleles of the same. So, and the biochemical mechanism and genetics of resistance development is being elucidated. So, how to tackle this? Now, this is an example of some of the pests which have developed resistance to BT, and the mechanism is again different. Some is by alteration in these ditoxin receptors. So, as I said, these are gadolinium alkaline phosphatase or aminopeptidase type of receptors, or ABCC transporters, or alteration in these proteases. So activation level is altered. So this ditoxin will not be activated. So if it not be activated, it will not bind to it. Or there will be sequestration mechanisms, that is holding up of toxin by in these glycolipid moieties, or the general immune response element. So this is just a basic slide to show the mechanism of resistance development at different levels. So at the activation level, at the receptor binding level, or at the cell immunity, cell repair level. And this is a detailed slide, so I will not go into details, but just to show that different mechanisms exist, either for alteration in the binding site of receptors or elevated immune response such that there is decreased growth. Now, the strategies to tackle these resistance, they have multiple strategies, and we call them as resistance management strategies. So they are at the level of either promoter. So we, instead of using a constitutive promoter, that is, which expresses the gene all through, we use either an inducible promoter, which is be expressed only upon some kind of induction, either wound induction or chemically induced. For example, salicylic acid, chemically induced promoter is available, or expression not in all plant parts, but only tissues. For example, for pigeon pea and chickpea, we need expression only in the pod, and for cotton bollworm, we need expression only in the cotton ball. So, or use different kinds of genes. So we use novel genes, or we use fused genes, that is using two different kinds of genes, fusing genes, and or we introduce more than one gene in the transgenic plant, which are having different receptors. So this is what we call as gene pyramiding. Or we use a very high dose of gene so that all the insects are free, nothing survives. So no resistant insects. Or we use the cultural practices, that is, we use refugees. So I will explain this. What are these refugees? So refugees basically reduce the selection. That is, we also grow non-BT crops. So now the current norm is that at least 20% of the field should be planted with non-BT crop. So that is, the insect is not only feeding on BT transgene, it also gets to feed on the non-BT crop, which is not having that gene. So the insect will be moving between these two, so the selection pressure. So this is a norm for growing BT crops. That is what I said, that on-farm monitoring is also important, that whether farmers are growing the non-BT, that is the control. Now, in Indian scenario, we are helped by that there are multiple cropping situations. So crops with different transgenes, which can serve as refugees to each other, and also a crop diversification. So we grow different types of crops, so that also reduces the selection. That is, for example, you are growing BT cotton and in between doing some vegetable also, so that is crop diversification. So spatial and temporal control, that is tissue specific, and time controlled expression. So inducible promoters are used, and so this leads to reduction in selection. So these are the approaches for minimizing survival, that we use gene pyramiding, that is, we use toxic proteins that bind to different sites in insect gut, or we use such high dose that it is the resistant population is unable to survive. So this is a little difficult to achieve, get toxins of that high. So, but still we can meet these objectives by, so we keep isolating for novel insecticidal genes. So this is the project I'm working on, and we do modification of the dry genes and construction of hybrid genes. So this slide is to show the synergistic behavior of these two different types of genes which have different receptor binding sites, the BIC type and the crystal proteins. And it is actually found that using these two types together, it also leads to some kind of synergy. So how we can construct the novel genes, which is will be based on changing some of the amino acids in the specific domains by site-directed mutagenesis and seeing effect on toxicity, or else changing the domain, taking one domain from one tritoxin and taking domain 2 from another tritoxin and so on. And so basically we want to have improvement in protein stability, receptor binding, and this pore formation and overall insecticide. So this is an example to show how some of these changes are done by site-directed mutagenesis. For example, in this one, there are changes in this domain 2 so that it led to increase in receptor binding. And I'm sorry, this is the slide in domain 2, which led to increase in receptor binding. And these are the changes in domain 1, which led to increase in pore formation. So now these are the different kinds of hybrid genes which have been constructed either through domain exchange or by site-directed mutagenesis, and which exhibited enhanced insecticide. So again, for these chimeric WIP proteins, in combination with dry genes, which are having enhanced and the effect on toxicity. So all these researches, they go on simultaneously in our search for looking for better genes. So constructed either through domain swapping or through site-directed mutagenesis. And now currently, this is the list of which are approved events. So as I said, the approval of commercialization for transgenic plants is done on a case-by-case basis. And each transgenic crop which has to be commercialized, that the one which we call it as an event. So we call these as events. So they are labeled as events. So even, for example, this one, this is the one which the developer is Syngenta, we give it a wheat cotton, and which contains this wheat pot. So we can see that now, right now, there is a combination of these different WIP and tri-type genes for commercial. And some of the transgenic plants are having both herbicide tolerance and insecticide resistance. So again, these are the which are having these tri and WIP genes. These are the events, and these are the trade names and the type of genes which they have. Some are having up to 3 different type of tri genes. Some are the modified tri genes, and they are having resistance. So now, how do we look for novel genes in BT isolate? BT has a wide distribution, and it is ubiquitous. So it is found in different soil, in water, in red insects, in phytoplankton, in green dust, and so on. Now, what should be the desirable attributes? That it should have novel toxic genes, and it should have high insecticidal activity and broader insecticidal spectrum, and toxins having different binding sites. So we isolate these WIP genes, and how do we construct novel genes? So we isolate, and how do we construct novel genes? We do it by protein engineering. So we take different domains. For example, we take domain 1 from tri1, 1 from tri2, 1 from tri3, and we construct these different hybrid genes by immunity. And now, how the search for novel insecticidal genes has proceeded. So we see a timeline for this. So there has been advances in gene isolation methods. And so as I said, the first gene was isolated by the Schneff and Whiteley in 1951, tri-gene. And since then, the initial genes were by hybridization. So then hybridization, that is rather cumbersome. Then PCR methods have been devised, which are very rapid and very easy to do. And then modifications of PCRs have been done over the decade of 19th and this millennium to isolate different novel genes. And now in this 2009 onwards, next generation sequencing has also been used for isolation of novel genes. So This is just a flow chart for how one would isolate a gene from BT. So we isolate BT from diverse samples and we molecular characterize these isolates and we do it with 16R RNA sequencing. So our RNA is short for ribosomal RNA. These 16 as ribosomal RNA sequences are highly conserved, by which I mean they will be same for a species. So if you want to know that this bacterium is Basilar stringensis, we do 16R RNA sequencing and we can know that it is. And then we determine what are the different kinds of dry genes present in it, or deep genes present in it by PCR screening. Then we amplify, clone, and do sequence analysis, and we express it in coli, and we test its toxicity. And if we want to develop a transgenic plant, we do codon optimization, get a synthetic gene, and develop the transgenic. Now, alternatively, one can also go into the route of evaluation of toxicity of BT isolates themselves and then select isolates with high toxicity. This is to show what I meant by regions, highly conserved regions, which are present. The green, these ones, 9 conserved regions which are present in bacteria and specific to each species. So by this, you can which is the bacterium. So this is a slide to show that one of the BT isolates, which we BT isolates from Ladakh, we isolated. So India, and these are the spores and crystals, and we did dot hybridization to know that Tri1 gene was present. And now BT has what we call as megaplasmids. That is, they are high molecular weight plasmids. So this is showing how we see the plasmid DNA on the gene. And for PCR, we design different kinds of primers for different genes. And these are the genes which we have isolated, and we subsequently deposit them to this NCBI gene bank. And this is what I mean by sequence comparison. So I'm just showing one slide for illustration. That we come, so the ours was this gene, right to AF. So this was AF1 was known previously, so we compared sequence with it. And the other gene we isolated was 2AB16. So we see wherein our gene in these boxes is different from the known sequence. So and we have, so this is just to show some of the work from my lab. We have isolated these genes and deposited them to, this is a culture collection in India, which is known as National Agriculturally Important Microculture Collection. It is in Mau, and we get an accession number for these. And as I said, 16 as our RNA accession number from NCBI Gene Bank. And we isolated these genes, and these are the accession numbers for those, and we are talking. Now we have also constructed synthetic genes, which are plant-preferred codon optimized genes, and we have developed tobacco transgenics to see their implantar efficacy. And this we do by cloning in this plant transformation vectors, developing transgenic plants, and then testing by PCR whether the gene is present, testing insecticidal activity, seeing histopathological effects in the insect vector, and testing level of mRNA by QRT PCR and bioassays and certain hybridization to see that the gene is integrated in the genome. And we saw that this gene were effective in lambda. Now, this is just to illustrate that now the whole genome sequencing is being done in a big way for bacteria as well as for crops. So this is how we do sequencing. We get sequences and we assemble into a genome sequence. We have also done this strategy, and for this we have done this full deconvolution strategy. So what we did was we combined megaplasmid DNA from 36 isolates, and we designed the strategy so that we sequenced this and we identified novel kinds of genes in this. Then we designed primers and we amplified these genes, and these were the novel genes we cloned from these isolates. These are some of the more promising dry and big genes which we have isolated, and that they are toxic to Parmigiana and also toxic to some of the aphids. And this is how we do sequence comparison. For example, this was with AAA1, which is the first gene which was isolated in 1996. And these are our genes. So we see there they are having these amino acid substitutions at different places. So we group them into 6 different phylogenetic groups, and we test. This is the cloning part, and we tested their efficacy on Helicobacter aeruginosa, and we're seeing that. So again, as I said, we do structural elucidation to see what could be the probable receptor binding sites. And for this, I am showing this example of this novel site 1027 protein, which we isolated. And we saw that it has these 5 motifs, which are also present in other cytolytic proteins. And we did this homology modeling, and we defined its receptor sites. And we are also in the process of constructing these Tri1AC and with the 44 fusion gene to see that if this fused gene will have toxicity. So right now we are doing modeling and seeing docking with receptor binding. Now these are some of the other insecticidal proteins apart from BT. But these have not been used as much. So there were some reports, but the BT transgenic crops are the most favored, and they are the ones which have been used. So one example is of this Photogrhabdus luminous bacterium. So it has these insecticidal proteins, which is also having was tested in this Arabidopsis thaliana, which is a model plant for transgenic expression, and it was seen to give protection from this medical sexta. The other is this enzyme cholesterol oxidase from bacteria, but again, this was having protection from ball weevil, but again, there hasn't been subsequent development of this. So these are my concluding remarks that there is a need for transplant. By it is estimated that by 2050, we will have to have 70% more food. So how do we get it? We get it through agricultural production innovations and biotechnologies. And this is again a slide to show the contribution of biotech crops to food security, sustainability, climate change, and solutions. So climate change solutions. So this shows that to provide a better environment and to alleviate poverty and hunger, need biotech. So what is the path ahead for us? So the path ahead is there is enormous potential for isolation of novel lipids, and I'll explain some of the approaches that are being used. And the creation of new type of glycoproteins is possible by designing based on molecular modeling. And for this, we also need continuous analysis of what is the molecular basis of toxin to better understand the insect specificity and how the insects develop resistance and what is the role of insects. And insect resistant transgenic plants with gene parameters. As I showed, that now up to 3 genes are being added in the new transgenic plants that are available. So we call single gene, we call first generation transgenic plants. 2 genes we call second generation, and now right now we are having 3 third generation transgenic, which are having 3 lines of genes. And so now along with this, we need to have accurate and adequate risk assessment and management. So this will also lead to greater public awareness, and we also need public confidence that transgenic crops are safe for consumption and for cultivation. And at the same time, we need to do all this in an integrated pest management strategy. That is, we also need to use other forms of biological control and also improve the farm practices, which leads to a better insect pest control in a sustainable way. I will end my talk here, and these are my PhD and MSc students, some of whose work I have shown, and my associates in this research. Thank you.
Thank you, Doctor, for your wonderful presentation. The session is open for discussion. Students, if you need any clarification, you can ask Dr. Sarjeet. Any questions? You can also use the chat box to ask questions.
Seems there are no questions, so we can end the session.
Some questions are coming.
The first is, what are the challenges the engineered crops for, I think, for tree, I think, for achieving stress tolerance. So some of these I have already explained. Is edible vaccine also somewhat related to this concept of transgenic plants? So edible vaccines are being developed, but I have not digressed into that, since that was, so I mainly dealt with biotic stress tolerance in crop plants. But yes, it is possible, and these are being developed, edible vaccine transgenic. What are the major criticisms of BT cotton by NGOs and concerns expressed by farmers? So some of these also I have addressed, the perceived risk. What are the concerns by farmers? So basically, one thing I would like to say here is that in India, the only plant which has been commercialized is BT cotton. And for BT brinjal, which has been developed, there is a moratorium since 2010. And in that context, public debates were organized, and it is put on moratorium. Although, as I said, BT brinjal has been approved for commercial cultivation in our neighboring country, in Bangladesh. And in BT cotton, the good thing in India is that the BT cotton is commercially released only as the hybrids. So hybrids, there the hybrids have to be, the farmers will need to have the fresh seed for every season. Otherwise, the efficacy will go up. So that is the good part in cotton. So in India, the BT cotton is available only in the form of cotton hybrids, that is taking the BT gene and introgressing it, that is crossing it with a local variety, so that one gets the advantage of all the locally adapted variety, which is good for that particular agroclimatic region, and plus the advantage. What pests are affected by BT technology? Some examples. So this also I have said that mostly the cryotoxins which have been used, they are, this was one of the initial slides, that they are good for lepidopteran, dipteran, polyopteran, hemipteran, and several other insect orders also. And the good thing is they are not toxic to humans and also to other vertebrates. So there is a vast range of insect orders, vast range of insect pests for which we can use these cryotoxins. What difficulties observed during production of BT transgenic crops? So this is a kind of broad question. What would exactly one mean is whether the difficulties during the development. So the process for the standardization would have to be done for development of that particular transgenic crop. That is why, as I showed in my slide also, sometimes the BT gene is first tested in a model plant. By model plant, we mean the plant which is easy to transform. For example, tobacco is one model plant and another is Arabidopsis thaliana. So many of the genes, apart from BT also, any transgenic gene, we test first in a plant which is easy to transform. And now the challenge or the difficulty will be whether that crop itself can be transform. It is amenable to genetic transformation. For example, wheat is difficult to transform, chickpea is difficult to transform, pigeon pea was difficult to transform, but in our institute it has been done. So that is one, that whether the plant can be transformed. That is one difficulty. You need to have process optimization for that. And then secondly, what will be the level of expression? So all those that slide in respect of transgenic tobacco crop in our lab, I show, that is done to see what will be the level, whether it will be, we call it implanter efficacy, to determine whether the plant will be effective, the gene will be effective in planter. At present, what are the other examples of other eatables at which work is going on for genetic modification so that they are more nutritive? So one is about golden rice, in which this work for what is developed. So and now in many public sector institutions, different kinds of genes are being used for developing transgenic plants, but because of stringent regulatory regime, and since BT brinjal, which was to be the edible transgenic crop, there is a moratorium. This research, although it is going on, the future is not clear that when they will see commercialization. There is GM mustard also, genetically modified mustard also developed by Delhi University. So the plate is hanging in balance that how they will, when they will be released. Other than brinjal, cotton, tomato, and golden rice. So this is basically a little broad question. I do not understand. Research on many crops is going on. So research on oil seeds is going on. A lot of research on mustard is going on cereals, rice and wheat and maize research is going on. So right now our national priority is basically increasing the oil production. So research on oil seeds is going on. Also, another priority is to use alternative cereals, which are having low glycemic index. So you also might also be knowing that for these days, this multi-grain cereals are being used, which are having ragi, barley, and so on. So research on those plants is also. Okay, I think we can end here. Thank you. Thank you. You have been a great audience. Thank you and all the best.
Thank you, Doctor, so much for joining us today.
Thank you. Thank you. Thank you, Dr. So may I leave now?
Yes, Doctor.
Thank you.
Thank you, students, for joining us today. We end our sessions today. Further announcements, I will give it in the WhatsApp group. Okay. Thank you so much.
Sir, excuse me.